Hello everyone, I am Dr. Sirajul Faizi. I welcome you in my YouTube channel. Today in this video, I am going to discuss about HS analysis. This is only for learning purpose. This video is only for learning purpose. Okay, what is HS analysis? Actually, this content from a paper, what is the HS analysis, how you apply in your research this analysis. You know, my friend, the concept of HS analysis is evolved from an attempt to specify the space occupied by a molecule. Specify the space occupied by a molecule in a crystal in order to divide the electron density of the crystal into molecular fragments of electrons density. You know, a Hirschfeld surface investigation or HS investigation is an effective way to quantify the nature of the intermolecular interactions. What does it mean? You quantify the nature of the intermolecular interaction. The Hirschfeld surface computations were done by the use of Crystal Explorer, from where you will get you have this Crystal Explorer, which represent the Hirschfeld surface plotted over a property named as D norm. This is the D norm. So this is the writing style. You also remember D is italic and on. In the range from red to blue. This is the references. Color coding is used in order to show intermolecular contact. Red, white and blue spots show intermolecular contacts with distances less than, equal to and larger than what was radii, respectively. The dark red regions around the protonated amino group of cation and one of the oxygen atom of carbonyl group of the anion indicate the presence of the NHO, this type of interaction. Here also H and oxygen, there is a dotted line, 3 dotted line, it is interaction in the crystal bag. Whereas the comparatively light red spot on HS means Hirschfeld surface indicate the CHO interaction. Not only hydrogen bond interaction can be explored by Hirschfeld surface analysis, but also comparatively weak interaction involved phenyl ring can be explored. The triangular shaped regions on the HS plotted over the shape index, I will show you the figure, around the phenyl ring indicate the pi-pi stacking interaction are present in the crystal bag. But it is found that the pi-pi stacking interaction is very weak, as the centroid to centroid separation range from 4.755 to 5.963 angstrom. You can see this is the range. The donor and acceptor groups can be classified by plotting the Hirschfeld's over electrostatic potential. Red and blue region on the HS represent the hydrogen bond acceptor and donor respectively. To view of Hirschfeld surface, I will show you. The blue spot on the Hirschfeld near the protonated amino group represent that it acts as a hydrogen bond acceptor, whereas the red spot on HS close to one of the oxygen atom of carbonyl group indicates that it acts as a hydrogen bond donor. So look at these figures. This is A and this is B. Here HS plot over D norm, and this one is shape index for any compounds, because this is the paper. So if you work for your compound, then this one will you have to write D norm, and this one is shape index. You know, my friend, further two-dimensional 2D fingerprint, FP fingerprint, and 2D means two-dimensional. Plot are used to provide quantitative information related to nature and type of intermolecular contacts experienced by the molecules in the crystal pack. 2D FB means fingerprint plots are computed for each interatomic contact and for overall interaction. In the computation of the individual interatomic contacts, the reciprocal contact of each interatomic contact is also included. So, you know, the overall interaction of this plot, spikes represent the interactions. I will show you what is the meaning of spike. SPIKE, spikes. Represent the interatomic contact that have the significant contribution to crystal packing. DI, and DI stands for distance from the Hirschfeld surface to the nearest nucleus inside the Hirschfeld, and DE, whereas DE stands for the distance from the edges to the nearest nucleus outside the edges. The interatomic contacts that have stronger contribution to the crystal packing as compared to other interatomic contact like HH, CH and OH with percentage contribution of 53.8 and so on. This is the, if you will do, you will get your values. So other interatomic contact that have comparatively less contribution to the crystal packing, so you have to write like this. So this video is actually I am making to understand little bit what is the results and what is the meaning of some terminology like D, norm, shape index, DI, DE, what is the FP fingerprint. Now, you know, this is the spike, and this spike giving some information. This is the HH interaction. You can see the differences. This is the CH interaction. So you can see this is the OH interaction. This spike is different. And CC interaction, you can see. NH interaction, you can see. OH interaction, OC interaction, and NC interaction like this. And this graph is DE versus DI. So interaction of all atoms represent inside the edges to an atom present in the vicinity of the edges is explored. This investigation inferred that all the edges contained atoms interact strongly with the hydrogen atom located in the vicinity of the Hirschfeld surface, and the percentage contribution of the all H interaction is 76. Other such interactions are all C, all O and all N with percentage contributions respectively. So these interactions are important to discuss. They help better understanding the crystal packing. Similarly, the interaction of an atom present inside the Hirschfeld with all the atoms located in the vicinity of the edges is computed to further explore crystal packing. This investigation inferred that hydrogen atom present inside the edges has strongly interacted with the atom of neighboring molecules present as compared to the other atom, the percentage contribution of particle. So you have to make this type of figures. So my friends, I think you understand little bit about HS, Hirschfeld surface analysis. If you like such type of video, then subscribe my channel, and thanks for watching.